Hello friends, my name is Deepak Kaldhone and you are welcome to our channel Forensic Search. Friends, in this video we will discuss one question paper of scientific officer in DNA section of Forensic Science Laboratory. This question paper is of year 2016 of Himachal Pradesh Forensic Science Laboratory. In this video, we will discuss some questions of forensic science which was asked in the scientific officer examination in DNA section. So, before going to the main video, there is one announcement is online coaching is available. That is, we are also providing the coaching for scientific officer post of following sections in forensic science laboratory. First one is biology and DNA section. Second one is chemistry, narcotics, toxicology section. Third one is firearm and physics section. Next one is question document section. So all the syllabus of forensic science in the above mentioned sections will be covered. If anybody is interested for online coaching, then such candidates may contact on this number. Now see the question paper of scientific officer in DNA section of 2016. In this question paper, we will discuss some questions of forensic science. There are total 100 questions and the maximum marks is 100 and time duration is 2 hours. Let's start with the first question. The first question is, who advocated first the application of science to criminal investigation? The first 30 questions are from the forensic science syllabus. तो उसमें से पहला क्वेश्चन है, who advocated first the application of science to criminal investigation? Option E is Alfonso Bertillon, option B Hans Gross, option C Paul Crick and option D August Ulmer. तो जिसको कोई आंसर आता है वो मेरे साथ साथ सॉल्व कर सकता है क्वेश्चन पेपर को. The correct answer of this question is option B, that is Hans Gross first advocated the application of science to criminal investigation. Then see the next question. In India, the first state forensic science laboratory was established at dash dash. Option A Calcutta 1952, option B Calcutta 1955, option C Delhi 1952 and option D Delhi 1955. The correct option of this question is option A, that is Calcutta in 1952. Then next question is, who propounded the principle of exchange? Option A Francis Galton, option B Edmund Locard, option C Hans Gross and option D Alfonso Bertillon. The correct answer of this question is option B Edmund Locard. We all know the Edmund Locard gives the famous principle known as principle of exchange, and this is the base of the forensic science. Francis Galton gives about the fingerprint, then Hans Gross अभी हमने देखा था, he advocated the application of science for the criminal investigation, and Alfonso Bertillon gives the science of anthropometry, that means the measurement of the body parts. Then question number 4 is, the most reliable form of evidence is option A blood, option B weapon, option C clothes, option D eyewitness. The correct answer of this question is option A, that is blood. Blood is most reliable form of evidence because the composition of blood does not change, and on the basis of the blood group and DNA we can definitely link the suspect of the criminal. That's why the option A is correct one. Then next question is question number 5, that is the final breakthrough for the fingerprint method for personal identification was made by option A Edward Henry, option B Sir Francis Galton, option C Joseph Reed and option D William Herschel. So the correct answer of this question is Sir Francis Galton. Then the next question is, quadrant or zone method of crime scene investigation is suitable for option A indoor crime scene, option B outdoor crime scene, option C automobile crime scene and option D none of these. The correct answer is outdoor crime scene. This quadrant zone method is useful for the outdoor crime scene. Then question number 7 is the acid phosphatase screening test is used to detect which physiological fluid. Option A sweat, option B semen, option C blood, option D saliva. So correct option for this question is option B semen, because in semen there is one enzyme known as acid phosphatase, and for the detection of this acid phosphatase screening test is used for this semen. Then question number 8 and question number 9 is of chemistry, pure chemistry type of question. Then question number 10 is which of the following individuals is known as the father of forensic toxicology? Option A R. Arias, option B Edmund Locard, option C Kelvin Goddard, option D Matthew Orpila. The correct answer of this question, that is father of forensic toxicology, is known as Matthew Orpila. So correct answer is option D. Edmund Locard gives principle of exchange. Then Kelvin Goddard is related to forensic ballistics. Then the next question is a scalp hair specimen with significant root sheet material would indicate that the hair originated from which phase of growth? Option A anagen, option B collagen, option C catagen, and option D telogen. The correct answer is option A, that is anagen. Then the next question is which of the following is prepared way to identify a deceased person? Deceased person means dead person. First option, that is option A, is visual inspection, option B tattoos, option C surgical scars, option D is examination of teeth and surgical records. The correct option for this question is option D, that is examination of teeth and surgical records. On the basis of the teeth structure and surgical records, we can identify a dead person. Then next question is which of the following best defines rigor mortis? Option A, this is the body cooling after death. Then option B, liver mortis. Option C, post-mortem rigidity. And option D, post-mortem medical examination. Okay, this question is from the medicine, forensic medicine. So which of the following best defines rigor mortis? So what is mean by rigor mortis? The rigor mortis is also known as post-mortem rigidity. That means, what is post-mortem rigidity? That means the stiffness of the body muscles. Rigor mortis or post-mortem rigidity is known as stiffness of the body muscles. Then यहाँ पे first option है, that is option A, this is the body cooling after death. Cooling of the body after death is known as rigor mortis. ALGOR, rigor mortis. ठीक है, mortis MORTIS. Rigor mortis means cooling of the body. Then liver mortis. Liver mortis is known as post-mortem lividity. That means what is post-mortem lividity? That means settling of the blood. जो भी बॉडी का पार्ट जमीन के कांटेक्ट में होता है वहां पे सारा ब्लड सेट हो जाता है, इकट्ठा होता है, उसको हम लिवर मोर्टिस और क्या बोलते हैं, पोस्टमार्टम लिविडिटी. This postmortem rigidity is also known as rigor mortis. That means stiffness of the muscles. And option D, that is postmortem medical examination, is known as autopsy or postmortem. Then the next question is which of the following physiological fluids will be expected to have a high concentration of the enzyme amylase enzyme? Option A urine, option B sweat, option C semen, option D saliva. So enzyme amylase is present in the saliva. That's why the correct answer of this question is option D. Then friends, we are providing the online coaching for the preparation of this scientific officer post. If anybody is interested, then such candidates may contact on the given number in the first slide. Then see the next question, blood or buccal swab for DNA analysis are to be taken from any consensual partner having sex with the victim within dash dash hours following intercourse. कि ब्लड सैंपल्स और बकल स्वैब हम कब ले सकते हैं फॉर द डीएनए एनालिसिस. ऑप्शन ए 24 आवर्स, ऑप्शन बी 36 आवर्स, ऑप्शन सी 48 आवर्स, ऑप्शन डी 72 आवर्स. द करेक्ट आंसर इज ऑप्शन 24 आवर्स. इन बिटवीन 24 आवर्स अगर हम उसका बकल स्वैब्स और ब्लड सैंपल्स लेते हैं तो वहाँ पे कंटामिनेशन ऑफ एविडेंस होने के चांसेस बहुत कम होते हैं. दैट्स व्हाई वी कैन एक्यूरेटली फाइंड आउट द डीएनए ऑफ दैट पर्टिकुलर सस्पेक्ट. देन नेक्स्ट क्वेश्चन इज luminol reagent reacts with blood in the presence of peroxide to emit light by chemical process is known as. Option A fluorescence, option B chemiluminescence, option C coagulation, and option D illumination. The correct answer is option B, that is chemiluminescence. So in this luminol test, the bluish color is found in the presence of the blood, and this luminol reagent that will define the presence of blood. Then question number next 17, that is the condition characterized by the absence of sperm cells in the seminal fluid is known as. Option A hypospermia, option B hyperspermia, option C oligospermia, and option D azoospermia. The correct answer of this question is azoospermia. Azoospermia, or there is another word that is known as aspermia, that means absence of semen or absence of sperm. Oligospermia means the low sperm count. Then the next question 18 is the determination of whether or not a substance is blood is best made by the means of preliminary color tests such as the Kestel-Meyer color test, which use the chemical. That means in the preliminary test of the blood, there is one test known as Kestel-Meyer color test. In this test, which chemical is used? Option A P30, option B benzodiene, option C precipitin, and option D phenolphthalein. The correct answer of this question is phenolphthalein. This phenolphthalein test is also known as Kestel-Meyer color test. Then question number 19, corpora basal index is used to determine. Option A age, option B sex, option C race, and option D stature. Stature means height. The correct answer of this question is option B, that is this corpora basal index is used to determine the sex of a particular individual. Then the next question is properties of evidence that can be attributed to a common source with an extremely high degree of certainty is or are. Option A individual characteristics, option B referent characteristics, option C comparison characteristics, and option D class characteristics. So here what they call to a common source. Common source, that's why the correct answer of this question is option D, that is class characteristics. Class characteristics are common in a group of people. Okay, that's why it is a common source, means कि commonly present होगा, तो that's why it is known as class characteristics. Individually होगा तो उसमें सिर्फ one individual में होगा तो उसको हम individual characteristics बोलते हैं. Then next question is how much seminal fluid does the normal male release during an ejaculation? Option A 2.5 ounce, option B 6 pints, option C 1 milliliter, and option D 2.5 to 6 milliliters. The correct answer of this question is option D, that is 2.5 to 6 milliliters. Generally 1.5 to 6 or 1.5 to 5 milliliters sperm is ejaculated by a normal male release. Then the next question is for most fingerprint examiners, the chemical methods of choice for visualizing the latent print is option A silver nitrate, option B iodine, option C chlorate, option D neodymium. The correct answer of this question is option D, that is neodymium method is commonly used to visualize the latent fingerprint by most of the fingerprint examiners. Then the next question is the State Forensic Science Laboratory Shimla was established in option A 1986, option B 1987, option C 1988, and option 1989. The correct answer is option C, that is 1988. The State Forensic Science Laboratory Shimla was established at 13 December 1988, and then that laboratory was shifted to Junga in 1996. Then the next question is, chiloscopy is the study of option A lips, option B eyes, option C ears, and option D nose. The study of lips is known as chiloscopy. That's why the correct option is option A, that is lips. Then the next question is, in the ABO system, blood group O is characterized by the option A presence of antigen A and the absence of antigen B. Option B presence of both antigens A and antigen B. Option C presence of antigen O. And option D absence of both antigen A and antigen B. So the correct answer of this question is option D. In O blood group there is absence of both the antigens, that is antigen A and antigen B. Then the next question is, the ideal place to record the temperature of dead body is option A rectum, option B axilla, option C mouth, and option D groin. So the correct answer of this question is option A, that is rectum. In dead body we measure the temperature from this rectum. Then next question is, diatoms in bone marrow are seen in death due to option A strangulation, option B drowning, option C electrocution, and option D asphyxia. So diatoms in bone marrow, ये किस में दिखता है हमें? डेथ ड्यू टू ड्राउनिंग. सो करेक्ट आंसर इज ऑप्शन बी ड्राउनिंग. ठीक है, ड्राउनिंग मीन्स जो पानी में डूब के डेथ हो जाता है उसको हम ड्राउनिंग बोलते हैं. एंड दिस डायटम्स आर द माइक्रोस्कोपिक ऑर्गेनिज्म प्रेजेंस इन द वाटर बॉडीज. सो दीज आर फाउंड इन द बोन मैरो. देन एस्फिक्सिया मीन्स डेथ ड्यू टू लैक ऑफ ऑक्सीजन. इलेक्ट्रोक्यूशन मीन्स जो इलेक्ट्रिसिटी होता है उसके कारण, शॉक लगने के कारण जो डेथ होता है वो. एंड स्ट्रैंगुलेशन बाय एनी रोप, कोई पर्सन किसी का मर्डर करता है तो उसको स्ट्रैंगुलेशन बोला जाता है. देन नेक्स्ट वन इज, स्पेसिमेंस फॉर टॉक्सिकोलॉजिकल इवैल्यूएशन शुड बी प्रिजर्व्ड इन ऑप्शन ए 10% फॉर्मल्डिहाइड, ऑप्शन बी अल्कोहल, ऑप्शन सी नॉर्मल सलाइन, ऑप्शन डी सैचुरेटेड सॉल्यूशन ऑफ कॉमन सॉल्ट. So the specimens for toxicological evaluation, that means the presence of poison or not, for this these specimens will be preserved in alcohol, or there is an alternative that is known as ethanol or isopropyl alcohol. So the correct answer is option B. Then the next question is, all of the following are items to be collected from a deceased body and sent to the forensic laboratory except option A head and pubic hairs, option B ocular fluid, option C blood, and option D fingernail scrapings. So यहाँ पे पूछा गया एक्सेप्ट कि कौन से के अलावा हम सारे आइटम्स टू बी कलेक्टेड फ्रॉम डेड डिसीज बॉडी और डेड बॉडी एंड इट इज सेंट टू द फोरेंसिक साइंस लैबोरेटरी. हेड एंड pubic hairs, then ocular fluid, then option C blood, and option D fingernail scraping. So except head and pubic hair, because ocular fluid जो कि आइज में प्रेजेंट होता है तो उसको हम सेंड कर सकते हैं टू डिटरमाइन द टाइम सिंस डेट. देन ऑन द बेसिस ऑफ द ब्लड वी कैन आइडेंटिफाई द ब्लड ग्रुप एज वेल एज DNA. एंड इन द फिंगरनेल स्क्रैपिंग्स मीन्स कि नेल्स के अंदर जो भी कुछ मटेरियल होगा, स्किन मटेरियल वगैरह, तो उससे हम कोई अगर सस्पेक्ट है जिसने उसका मर्डर किया होगा तो वी कैन आइडेंटिफाई दैट पर्सन. ठीक है. हेड एंड pubic hair, हेड ईयर भी इंपॉर्टेंट है, बट हेड कभी कभी हमें मिल सकता है, नहीं मीन्स कि हेड के ऊपर हेयर्स मिल सकते हैं, नहीं मिल सकते हैं. एंड ऑन द बेसिस ऑफ द हेयर वी कैन आइडेंटिफाई द DNA. तो इससे बेटर है कि वो ब्लड से भी मिलता है, तो वी कैन सेंड द ब्लड सैंपल, ocular fluid एंड fingernail scraping. तो इसकी हमें ज़्यादा जरूरत नहीं होती. देन नेक्स्ट क्वेश्चन इज, based on a mathematical calculation by Victor Balthazar, the probability of two individuals having the same fingerprint is 1 out of option A this one, option B, then option C and option D. That means यह क्वेश्चन में क्या पूछा गया कि जनरली फिंगरप्रिंट्स किसी भी दो पर्सन्स के सेम नहीं होते, तो यहाँ पे एक प्रोबेबिलिटी दिया गया है कि टू इंडिविजुअल्स का फिंगरप्रिंट सेम हो सकता है. तो वो फिगर कौन सा है? करेक्ट आंसर है ऑप्शन डी, दैट इज 1 into 10 raised to 19 power. ठीक है, 10 raised to 19 powers means अभी तक अपनी इतनी population नहीं है. That's why there is a no chance to having the same fingerprints of two individuals. That's why this is the correct answer given by this Victor Balls hazards. So friends, these are some important questions of forensic science which were asked in the scientific officers of DNA section in forensic science laboratory. We are also providing the coaching of the forensic science syllabus. If anybody is interested, then such candidates may contact on that given number. Thanks for watching our channel Forensic Search. Thank you.